So in today's lecture, we'll be talking about the metabolism of high-density lipoprotein. The HDL particle has the smallest amount of triglycerides, and because of that, it is the smallest in size. However, a larger fraction of this HDL particle is made up of proteins, and because of this large amount of protein that it contains, it has the highest density. So compared to other lipoproteins, the HDL particle is the smallest in size with the largest density. Chylomicrons, very low density lipoprotein, and low density lipoprotein. They carry cholesterol from the intestine or from the liver to extrahepatic tissues. And because of that, high amount of these lipoproteins in the circulation poses an increased risk of developing atherosclerosis or cardiovascular diseases. On the other hand, high density lipoprotein plays a role in reverse cholesterol transport, in which it removes excess cholesterol, or it removes cholesterol from these various tissues, from these various extrahepatic tissues to the liver, where they are excreted in bile. And because of its ability to function in reverse cholesterol transport, high level of HDL in the circulation decreases the risk of developing cardiovascular diseases. So the HDL has several functions, which include it is anti-atherogenic, like I just mentioned, it is anti-thrombotic, and it is anti-inflammatory. Now, the metabolism of HDL occurs in three phases. The first is the formation of the HDL particle. The second is HDL cholesterol esterification, or esterification of the HDL cholesterol. And then the third one is the metabolism of HDL itself. The formation of the HDL particle begins with the synthesis of a type of protein called ApoA1. So we have a diagram here. This represents the intestine. This is the liver. These represent extrahepatic tissues, which include the cardiac muscle, the skeletal muscle, and the adipose tissue. And then this is the kidney. Also, this represents the blood circulation. This part of the diagram represents the blood circulation. So the formation of the HDL particle begins with the synthesis of a type of protein called ApoA1. So this ApoA1, or apolipoprotein A1, is predominantly synthesized by the liver and also the intestine. So this is ApoA1 being synthesized by the liver, and then this is ApoA1 being synthesized by the intestine. So whether it is being synthesized by the intestine or by the liver, it is released into the blood circulation. Now, in the blood circulation, they flow through various tissues and various organs and various cells. So as this ApoA1 flows through the capillaries of these extrahepatic tissues, free cholesterol and phospholipids will be pushed out, which will bind to the protein. Also, as the protein flows through the liver and the intestine, free cholesterol and phospholipids will also be pushed out from these various tissues and into the circulation, which will also bind to ApoA1. So here we see that this is ApoA1 binding. This C represents cholesterol. So this is basically free cholesterol. There's a transporter protein that ensures or facilitates the movement or the moving out of these free cholesterol and phospholipids from these various tissues. Okay? And that transporter protein is called ABCA1. So on the surface of the extrahepatic tissues, there is ABCA1. On the surface of the intestine or intestinal cells, there is ABCA1. And on the surface of the liver, there is ABCA1. So this ABCA1 facilitates the transport of free cholesterol and phospholipids from these various tissues into the circulation. And ABCA1 means ATP binding cassette A1. So this ATP binding cassette A1 is a transporter protein that facilitates free cholesterol and phospholipids to move out from the extrahepatic tissues, from the intestine, and from the liver into the circulation, which will bind to the ApoA1 in the blood, and that will result in the formation of the nascent HDL. The nascent HDL is also referred to as the immature HDL or pre-beta HDL. Now, the function of the ApoA1 is that it ensures the structural integrity of the HDL particle. Now, this is the nascent, this is immature. It is immature because this structure resembles or looks like a disc. So, ordinarily, lipoproteins are supposed to be spherical, but this resembles a disc rather than being spherical. And because it looks like a disc, it is referred to as the immature or the nascent high density lipoprotein, which is not functional. So, for the nascent HDL to become mature HDL, there must be the esterification of the HDL cholesterol. So the esterification of HDL cholesterol is facilitated by an enzyme called LCAT. LCAT means lecithin cholesterol acyl transferase. And this enzyme catalyzes the transfer of fatty acids from phospholipids to cholesterol. So this LCAT catalyzes the transfer of fatty acid, a fatty acid molecule from the phospholipid to cholesterol. You know, the HDL particle already has phospholipids together with the cholesterol, free cholesterol and the apoproteins. So there is a transfer of a fatty acid molecule from the phospholipid to the cholesterol, and such process is catalyzed by the LCAT. Now, the transfer of that fatty acid molecule to the cholesterol will cause the cholesterol to be esterified, and that will result in what? Cholesterol esters. So Ce here represents cholesterol ester. Now, the cholesterol ester is highly hydrophobic, and because it is highly hydrophobic, it cannot dissolve in the blood, and because of that, it will move into the core of the HDL particle. So once there is this esterification of the cholesterol esters will now move into the core of the HDL particle, and that will make the HDL particle to be larger in size and more spherical. So we can see this is larger in size compared to this and more spherical. So at this point, it is now referred to as the mature HDL, also refers to as alpha HDL. Now, the mature HDL will do either of two things. First, this is number one, and then this is number two. So the mature HDL can be taken up directly by the liver. Now, on the surface of the liver, there is a receptor called SROB1. SROB1 stands for scavenger receptor class B type 1. Scavenger receptor class B type 1. So this receptor has the ability to recognize and bind to the mature HDL. So number one, it is either this mature HDL will be taken up directly by the liver, where it will bind to this receptor. Once this mature HDL binds to this receptor, the HDL particle itself does not go into the liver. Rather, the cholesterol esters contained in its core, in the hydrophobic core, will now be released into the liver. That is number one. So the second one is that it can associate with circulating lipoproteins that contains ApoB on its surface, which can include the chylomicron or very low density lipoprotein. You have the chylomicron or very low density lipoprotein that contains ApoB on its surface. So what this mature HDL does is it gives cholesterol esters to these lipoproteins that contain ApoB on their surface, and then this chylomicron in turn gives triglyceride to the HDL particle. So HDL particle gives cholesterol esters to these lipoproteins, these circulating lipoproteins, while the circulating lipoprotein in turn gives triglyceride to HDL. And when that occurs, it will result in the formation of this, where the amount of triglycerides, you can see that triglyceride is larger than cholesterol esters, where the amount of triglycerides will be higher compared to that of cholesterol esters. And this also will be taken up by the liver. On binding to each specific receptor on the surface of the liver cells, the triglycerides will be hydrolyzed or be broken down by hepatic lipase. So in the liver, there is also lipase, an enzyme called lipase, which is referred to as hepatic lipase. That's HL, which represents hepatic lipase. So this hepatic lipase breaks down or hydrolyzes triglycerides to form fatty acids, which will now be absorbed by the liver. So whether the mature HDL particle chooses to be taken up directly by the liver or exchanging cholesterol esters for triglycerides to form a particle that is rich in triglycerides and then taken up by the liver and then be metabolized or hydrolyzed by hepatic lipase, it will result in the formation of HDL remnant. So whether the mature HDL follows this path or this path, it will result in formation of HDL remnant. And the HDL remnant consists of a little amount of cholesterol attached to the ApoA1, and then this will be carried by the blood to the kidneys, and then it will be filtered by the glomeruli, and then in the proximal convoluted tubule, it will be metabolized and reabsorbed back into the circulation. I stated earlier that ABCA1 is the transporter protein that ensures that free cholesterol and phospholipids from the extrahepatic tissues, intestine, and the liver are released into the circulation that will bind to ApoA1. So a genetic mutation of this transporter protein will affect its function, and that will make enough free cholesterol and phospholipids not to be pushed out of these various tissues into the circulation. And so what will happen? There will be accumulation of these fats. Okay, there will be accumulation of the free cholesterol and phospholipids in these various tissues, in the extrahepatic tissues, in the intestine, and in the liver. There will be accumulation because this is not functional. So if there is a mutation in this gene, it will not carry out its effect. And its effect is that it allows free cholesterol and phospholipids from these various tissues to be pushed out into the circulation. So once there is a defect, genetic defect of this gene, okay, or this transporter protein, so it means this free cholesterol and phospholipids will not be pushed out of these various tissues into the circulation. And so there will be accumulation. They will stay back, okay, they will be trapped in these various tissues, and that will result in the accumulation of free cholesterol and phospholipids in the various tissues. Also, there will be a decreased concentration of high density lipoprotein because these free cholesterol and phospholipids are what we bind to ApoA1 to form the HDL particle. So if they are not pushed out of these various tissues into the circulation, it means sufficient amount of HDL particle will not be produced. It means there will be a low amount of HDL in the blood, and then there will be also what? Accumulation of free cholesterol and phospholipids in the various tissues. An example of a disease in which there is a genetic defect or deficiency of this ABCA1 is Tanger's disease. So in Tanger's disease, there is deficiency of the functional capacity or functional ability of this gene. So basically there is there is a genetic mutation of this gene, and as a result of that, free cholesterol and phospholipids are not pushed out of these various tissues into the bloodstream, and that will cause the accumulation of free cholesterol and phospholipids in these various tissues, and then it will result in the decreased concentration of high density lipoprotein. I talked about an enzyme that converts this immature HDL to mature HDL by what? Catalyzing the esterification of cholesterol. So if there is deficiency of LCAT, What it also means is there will be accumulation of these fats, the free cholesterol and phospholipids in the various tissues. Also, there will be decreased concentration of high density lipoprotein. A genetic mutation of this transporter protein or deficiency of LCAT will result in the accumulation of free cholesterol and phospholipids in the extrahepatic tissues, in the intestine, and in the liver. And then that will result in decreased concentration of high density lipoprotein in the blood circulation. For example, we have a condition known as fish eye disease. Now, this fish eye disease is a condition where there is deficiency of LCAT. And because there is deficiency of LCAT, there will be accumulation of free cholesterol and phospholipids in the various tissues. And that will result in the deposition, okay, in the deposition of free cholesterol in the cornea of the eyes. And that will increase the opacity. So there will be corneal opacity. So in this disease condition, called fish eye disease, there is deficiency of the enzyme LCAT. And because there is deficiency of this enzyme, free cholesterol and phospholipids will be accumulated in the extrahepatic tissues, in the intestine, and in the liver. And that will result in deposition of free cholesterol in the cornea of the eyes, and that can increase the opacity. Okay, so that can result in corneal opacity, where the eyes will begin to resemble that of a fish, and that is why it is described as fish eye disease. This ApoA1 stimulates the action of LCAT. So LCAT stimulates the esterification or catalyzes the esterification of cholesterol, thereby converting the nascent HDL or the immature HDL to the mature HDL. However, its function is facilitated by ApoA1. So deficiency of ApoA1 can also affect the functional capacity of LCAT. So basically, this entails the metabolism of high density lipoprotein.